Welcome to day 4, working towards lab practical number 1 in Bio210. This is also a lesson that you'll see in your Bio114 class if you're in my Friday course. And so we'll take a look at the syllabus and see what's involved in today's activities. So on our 4th day, we have a new set of experiments, a whole section of experiments that are entitled Carbohydrate Catabolism and Fermentation. So we're going to be studying how bacteria utilize sugars both aerobically as well as anaerobically, and compare and contrast them with a couple of key experiments. One being OF glucose, which stands for oxidation fermentation of glucose. And we'll also be looking at one called phenol red broth. And the phenol red broth experiment and the OF glucose experiment will be set up, but I'll be lecturing on them and talking about their results during next laboratory period. Most of what we're going to spend our time on today, though, is going to be the streak plate and the gram stain, exercises 1-4 and then 3-7. So we're going to start straight away with the streak plate as a method for isolating bacteria from large groups of organisms in a collection like a tube or a Petri plate, and then streaking them out, systematically spreading them around so that we can isolate individual bacteria from very large groups in a sample. And then we'll end with the gram stain as another big lesson today. All right, we'll start with the streak plate. Okay, so if you look in your lab manual, the purpose of performing a so-called streak plate is that we want to be able to isolate individual bacterial cells from a group that they may have come in within a sample that contains thousands or millions of bacteria, and yet we want to be able to reach into that sample and pluck out one little organism and study it from the rest. It's also a way of determining maybe how many different types of organisms are in a given sample. So the streak plate has a purpose of isolating individual colonies of bacteria that are thought to have originated from one single cell that's been separated from a big sample. And then that sample may contain just large numbers of bacteria of all the same kind, or in most cases, especially if it's an environmental or a clinical sample, there could be a mixture of bacteria in a big population, and you're just trying to tease them apart. And we'll see that you can systematically, through a streak plate technique, spread them around so as to get individually isolated colonies after proper technique. All right, so we're going to be taking a sample in this case that's a couple of mils of liquid. This is a couple of milliliters, or here it says 1 milliliter. It won't matter. This particular sample could have been taken from anywhere. This is just a lab preparation to demonstrate how the technique works. But imagine if this was a blood sample, or maybe it was a urine sample, or any other kind of liquid sample, cerebrospinal fluid. I mean, pick your liquid that could have come from a person who could be showing signs and symptoms of an infection, and then a streak plate technique would be one particular starting way to try to tease apart, if the collection of bacteria in here is causing the infection, we need to begin to identify the relative numbers, and importantly, which kinds of genus and species could be infecting the person. This could also be a sample taken from the environment, so maybe some potentially dirty water and you want to know what's in it. Or maybe it's food, like milk, and you want to analyze which types of bacteria are in some dairy product, and so we could do this particular technique. And the end goal would be to make it look like this. Now, this particular picture taken from your book is pretty nice because it shows the bacteria in a red color. And I went out of my way today to put in some of those red bacteria. That organism is called Serratia marcescens. And although we don't need to memorize its name right now, it gives us a nice red color that can be differentiated from normal bacteria, which are typically just white or unpigmented. And just for fun, for challenge, I went ahead and I put in both types. So I have ordinary bacteria that would be clear and or white and uncolored, like E. coli. And mixed in there with the E. coli, I have some Serratia red bacteria. So we're going to see, if everything goes well, we're going to see a little bit of red and white bacteria on the same plate. All right. So how are we going to get this figured out? This is going to be our technique, and we're going to follow 4 different phases of streaking a plate, starting with starting with depositing the bacteria in quadrant 1, which becomes a lawn. So I'll pretend that this is my loop for now. Remember that the inoculating loop, or the loop that we grab the bacteria with and move bacteria around, is really small. And so just for the purposes of demonstrating real quick, before I use a real plate and go live, let's use the end of this. This is called a striker that we usually light the Bunsen burner with. But this end of it, we'll pretend that's our loop. And so I'll be showing you in just a few moments that you'll flame off your loop and then take the nice sterile loop that's cooled off and clean and put it into our sample of bacteria. And you'll just be getting a loop full, which is the amount of liquid that will fill up the inoculating loop. And then you'll carefully take that loop and wipe it onto the surface of the plate. And my figure shows that as one line. I'm actually going to flip the loop over, though, and just wipe it one more time. So for good measure, it'll be wiped off top and bottom. And then you'll simply take the loop and move it around, back and forth, overlapping streaks to occupy about maybe 20% of this part of the Petri plate. And that way you'll make a nice thick coverage of the lawn. You're trying to make it look like this section that we see here on the picture. And so in this step, it's fine to let the loop press flat onto the plate. You're making broad, smudgy streaks and trying to get good coverage there. Now, after you get done with that step, which is making quadrant 1, we're going to be then cleaning the loop off. So we'll flame the loop and we'll make a second set of streaks. When you go to make your second set of streaks with your nice clean loop, you want to use the loop slightly differently. If this is the surface of your Petri plate, you want the loop to actually stay at about a 30 to 45 degree angle. And you don't want it pressed flat like you did in the first quadrant. And the reason is because as you streak from side to side, let's say you're going from left to right, you pick up the loop and you make a streak. You have to imagine that you're wiping the bacteria off the bottom edge of the loop as you make that streak. And then you simply pick up your loop and you set it back down deep inside the lawn from quadrant 1, and you slide it across again. And you pick it up and you repeat this process at least 6 to 8 times so as to make some separate non-overlapping streaks by keeping the loop up at that angle and imagining you're wiping the bacteria off the edge of the loop. And what you're really doing is you're picking up bacteria from here and you're moving it across into this second quadrant area. Of course, the amount that we start with here will then start to thin out because of the way that we're using the loop. And we're flaming the excess amounts of bacteria off when we start each new quadrant. So when we get done with this second set of streaks, you'll see that the second set of streaks is thinner in density than the first set of streaks in the lawn. And then we're going to seek to make the third set of streaks just like the same technique as making the second set. You'll flame the loop off, and then you'll take your nice clean loop, hold it at a 30 to 45 degree angle, and start making your streaks from left to right, side to side. Important point, guys. If you're handling a loop side to side like this, that's the motion. You'd never want to push a loop. Sometimes I'll see students, they grab their loop and they push it forward. So if you were making these streaks, they'd start up here like this at this angle and push the loop forward. And you could imagine that if you do this, there's a risk that the loop could shovel underneath the auger, especially when auger is just this really soft jelly-like surface that then the sharpness of the loop could shovel under there. And if that happens, you've messed up and you'd have to start over. Again, the loop needs to go side to side. We also don't really want to pull the loop. Sometimes I see a student, many students make this mistake. They take the loop and they pull it, and the bacteria are trailing off the loop behind how they're pulling the loop. The problem here is that makes for a half moon shape. We do not want our streaks to have this half moon arc shape, which is too broad and too wide. What we're really looking for are nice, narrow lines, tight streaks. And so when you make that third set of streaks, you're going to make a half a dozen to maybe 8 streaks, just like you did in quadrant 2. And you will see that the growth in this quadrant will be even thinner. So looking at the third set of streaks, the end result will have maybe even some isolated colonies. It depends on how many bacteria you start with. Now, the fourth and final step, we're going to recommend that you do not flame the loop. So it says make a zigzag streak without flaming the loop. So in this case, you got to remember, just rotate the plate and make your zigzag streak at the end. If you accidentally flame the loop, it's still worth it to continue doing what you're supposed to do. But ideally, when we're working from a liquid sample like this, the concentration of bacteria would start to thin out if you flame your loop on that end process. And you might not get any growth in the fourth quadrant. It really depends on whether you're working from a liquid sample like that, or if you were working from a solid sample. You guys may recall that we made slants on one of our first, during our first week in doing experiments. And these slants, wait, did we make slants? We did, and I forgot. And those slants would be like working from a solid sample. If we went in and got a sample on the end of our loop there, then we might actually want to flame the loop because we're dealing with very concentrated sources of bacteria. So it depends on what you're working with. So the fourth set of streaks, you're still going to have your loop at a 30 to 45 degree angle, not flat. You'll start in the third quadrant and move some bacteria out like this, but then don't lift the loop. Just keep it on the surface of the auger and finish out with this zigzag set of streaks. The hardest part of this whole procedure is being able to see what you're doing, especially your first time at this. You've got to find a reflection off the incidental light in the room as it hits the auger surface. Everything you're doing is invisible, so it's very difficult sometimes for students to see, am I making these streaks effectively without touching back into the first quadrant, without having these streaks touch back into the second quadrant? We want to keep all the streaks situated and separate just as they're drawn in this picture. I'm going to go ahead and stop the camera for a moment, bring you guys back in with a zoomed in close-up look at this so you can see exactly how my hands are working to make an effective streak plate. All right, now we have a zoomed in close-up view so that you guys can see exactly how the streaking the plate will take place. First, I need to light up the flame. Okay, got the gas pressure right, flame is lit. And remember, when you have a flame that you're using for technique here with aseptic technique, you want to make sure that the flame is adjusted with the air holes open so that we can see the inner cone, the two different parts of the flame, including the tip of the inner cone, where we're going to work to make quick work of this loop here. So we can see when we flame sterilize the loop that it turns orangey hot. So now that loop is flame sterilized, and while it's cooling down, we can take our sample of mystery bacteria, and first of all, we'll just mix it up a little bit. We want to make sure that that's well mixed before we take the cap off, and then give it a quick flaming on the tube. You always flame it before you go in. And what I'll do is go in and just get a little loop full of bacteria. Okay, so I've got a loop full. And what I'll do from here is flip over my labeled plate. I've already got it labeled streak number 1. And I'm going to work near the flame. And I'll wipe off the loop and flip it over, and wipe it off again. And now I'll move that loop back and forth at a fairly low angle to make the lawn. And I'm not sure how well that can be seen in the close-up view right now, but just very lightly, I'm working the loop back and forth to make that lawn. Okay? So every time you move from one quadrant to the next, you want to make sure that you flame your loop. And the reason is we want to destroy all the excess bacteria so that every time we make a new set of streaks, we'll get fewer bacteria. That's the goal here, is to spread out the bacteria. So what I'm doing is I'm using the clamshell technique, which is a technique where you only lift the lid as far as necessary so that you can get access to the plate, but you're trying to reduce the chances of contamination falling in. Also, the open plate is only open near where the flame, where the Bunsen burner is. And the goal here is to reduce the chances that bacteria or fungi, whatever contaminants in the air won't get in. Okay, now I'm going to go ahead and make my second set of streaks. So I made 1, 2, 3, 4, 5, 6, 7, 8. So I made 8 parallel streaks from the lawn and into the second quadrant. And now I'm going to rotate the plate again. Now, beginning students and anybody who might just have a shaky hand in general, if your fine motor skills aren't too steady, what you want to do is use your elbow or your forearm to rest it on the counter and stabilize your loop. And also, you want to keep your wrist locked so that you're not drawing the lines, but you're actually pivoting from side to side with your wrist in that fashion so that you can make some straighter than average streaks here. And I always start by setting the loop down in a sterile part of the plate to make sure it's nice and cool before I touch those bacteria from the previous set of streaks. Okay, it's always a good habit. You don't want to fry the bacteria. If you go in there with a hot loop and you hear a sizzle, then, you know, on the microscopic level, you know what you're hearing. You're hearing, ah, and, you know, we don't want to kill those little guys. We're trying to keep them part of our experiment. So anyhow, all right. Now, I didn't flame the loop because I'm on my 4th set of streaks. So between the 3rd and 4th set of streaks, we do not flame the loop. I'll put the loop deep in the 3rd set of streaks, bring it over. And remember, I'm going to keep the loop down and do what's called a zigzag set of streaks. And eventually, I'll come out and then finish up like that. Okay, so that is how you streak a plate. And always remember, when you're finished with your work, to take your loop or whatever instrument and flame sterilize it so that it's safe to set back down. Now that I've streaked my plate, I'll put that in the incubator and we'll take a look at it next time. So hopefully I will have some nicely well isolated colonies similar to the picture that we see here. Okay, let's move on to a new activity. We'll look back at streak plate day 2 when we get there. Let's look at the gram stain. This is one of the most important lessons that you guys will learn all semester long. Aseptic technique might be lesson number 1 that everybody's here to learn, but the second most important would be the gram stain, just in terms of how common and how important it is as a part of micro as a subject, especially for healthcare professionals. So nurses and doctors and PAs, pharmacists, veterinarians, you name it, they're all going to need to know the meaning of the gram stain, and I want you guys to know how it's performed. Now here's a picture of a gram stain right here with 2 different types of bacteria present in the slide. We can see light pink cells and we can see dark purple cells. And those are the gram negative and gram positive cells. The gram negative cells, as they're referred to, are the light pink, and then the gram positive cells are dark purple. And we want to seek to understand why, and why is it called the gram stain anyway. So our purpose in performing the gram stain is to differentiate between so-called gram positive and gram negative bacteria, and then understand how that result is so important, guys. It says here that the gram stain, if you're not already interested, it's the single most important medical procedure in bacteriology. So when it comes to patient clinical samples, a gram stain is almost always ordered for anything that's bacterial in nature. So every day in hospitals across the globe, this gram stain is ordered day in and day out. And luckily, it only takes a few minutes to perform once you have a good sample to process, to put through the gram stain. And so within, say, 10 minutes or so, if you have the sample ready and you do the gram stain, then you can report back to someone very quickly, and this will allow a practicing physician to give good drug treatment, knowing the difference between whether the bacteria appear pink and therefore gram negative, or purple and then gram positive on the other hand, will allow the physician to not only take a big step in the right direction towards a genus and species identification. Is the patient infected with E. coli, which is gram negative, or maybe staph, which is gram positive? But also, then if you know you're treating E. coli, then that's going to guide appropriate antibiotics that are better at treating gram negatives. And likewise, the same for gram positives. You might have a different set of concerns pharmaceutically with the antibiotics that you'd be choosing. Now, let's dig back. Let's dig into the history just a little bit about the gram stain. I'll just jot a couple notes down here about its origins. Okay. In 1883, more than 100 years ago, right? We're not even talking last century. We're talking about the 1800s. There was a guy who came up with this procedure, not really knowing that he was setting out to change the world of medicine, but a man named Hans Christian Graham was a microbiologist, and he had friends who were doctors, and he would ask those doctors, he would say, hey, I would really like to look more closely and to study clinical samples that maybe have bacteria that are causing throat infection or skin infections or GI tract infections. And he knew that bacteria played a role in diseases that doctors were seeing, and he wanted to find a way to study them under the microscope and see what he could see. So Hans Christian Graham is famous for finding a unique and really informative way of looking at bacterial species, or at looking at different types of bacteria based on this staining combination that he came up with. So initially, Mr. Graham had some crystal violet available, and crystal violet is a purple dye, as the name implies, that is just soaked up by bacteria, just like you learned about in the simple stain. So it has a positive charge, and that will be absorbed by bacteria that have an opposite charge. If you recall, all cells have a net negative charge that will be able to absorb a simple staining dye like crystal violet. So, Graham had put that onto cells, and while he was looking at them under the microscope, he was able to see them better with the purple contrast in his microscope. But Graham was messing around with more than one type of chemical, and at some point he had applied a chemical that he called a decolorizer, and his decolorizer was almost pure ethanol. And ethanol is alcohol that can wash away dyes. So like if you get a stain in your clothing or something, alcohol might actually help as a solvent to dissolve some sorts of stains depending on what stains your clothing. And if you stain bacteria, like with crystal violet, he found that the alcohol that he had used on his stains would reduce the coloring, give some sort of reverse the staining process. Now, why he was doing that in the first place, I'm not exactly certain, but he noticed something really unusual when he applied the alcohol. He noticed that not all cell samples would decolorize at the same rate or with the same effectiveness. And so he started realizing that, for instance, staph bacteria that live on the skin or strep bacteria that live in the throat, predominantly those cells could retain the crystal violet purple coloring and actually, to a degree, resist the effects of the alcohol. And so he found that those bacteria had a tendency to be able to resist decolorization. And he compared that to bacteria that were commonly found in the gut, like E. coli. And if you're talking about E. coli by comparison, E. coli will readily soak up violet dye, but E. coli was not able to resist decolorization with an alcohol-based solvent solution. As soon as he washed those samples on his slide with the alcohol that he was using, the cells lost their color almost immediately. And so he started wondering if maybe there was something to that, if maybe there was a reason that he didn't understand that some cells would lose the crystal violet. But one of the things he came up with was that safranin, yet a different simple staining dye, could be absorbed by the cells that had lost the purple coloring. And so you could kind of counterstain or backfill, if you will, the cells that didn't have any coloring left because they're hard to see under the microscope. So he would add the safranin in to try to get the cells to absorb a light pink dye. And it sounds a little strange, like why would he put on crystal violet, which is a deep purple color? And what might happen if you put a light pink color on top of that? Because in theory, all the cells will absorb the dye. What he came to find out was that when you put light pink on top of deep purple, the deep purple cells stay deep purple. They're still violet colored. But the cells that had lost the purple coloring would readily take up the light pink dye and actually be more identifiable than if they had remained clear after decolorization. Now there's one more step to the development of Graham stains, to Graham's method to Graham staining, and what he ended up adding to the process is an iodine-based solution. So Graham's iodine is used right after putting on crystal violet, and we'll see in just a few short minutes how it is that that crystal violet and iodine makes for an enhanced staining technique so that the gram stain can truly give the results, the most accurate results that we would want. And so this whole thing was developed at such a time period where he noticed that there was a big difference between these different cell types, either ending up ultimately purple at the end of the process or pink, but he didn't understand why, other than just associating where those samples were taken from the body. And it turns out, underneath it all, at the molecular level, at the level of seeing these cells, even submicroscopically, these are diagrams that are based on pictures that were taken by electron microscopes. So an electron microscope, which has the ability to magnify images of real bacteria at millions of times their original size, allows these details to be possible. And that's very different than the light microscopes that we have in lab, where we can only magnify the bacteria about 1,000 times, right? So maximum magnification of 1000x will barely allow us to see the bacteria, but these drawings, this would be like millions of times their normal size. And what this diagram communicates is that gram-negative cells on the left, when you take a cross-section of a bacterial cell membrane and cell wall, is that you'll see the phospholipid bilayer that makes up the cell membrane, and that's talked about in lecture during chemistry, and that's present in both gram-positive and gram-negative cells. Now, if you recall back to lecture, in chapter 1, we learn about a special chemical called peptidoglycan. And peptidoglycan is specific to bacteria, whether they're gram-negative or gram-positive bacteria. Almost all bacteria have at least a little bit of peptidoglycan in their cell wall. And so when we look at gram-negative bacteria, we now know that there is a very thin layer of peptidoglycan. And when you contrast that with gram-positive bacteria, by comparison, they have a whole lot of peptidoglycan. So one of the things that we can take note of and that you guys need to memorize is that gram-negative bacteria have thin peptidoglycan. And by comparison, gram-positive bacteria have thick peptidoglycan. These diagrams are not drawn to perfect scale. And if they were drawn to perfect scale, we would have an issue here, because our textbook states that gram-positive bacteria have been found to have approximately 80 times more peptidoglycan thickness than their gram-negative counterparts. So if this was 80 times thicker than the layer that we see pictured here, this may as well just be like a pixel line across here, something that you wouldn't even notice was there. So do realize that the thinness, relatively speaking, of gram-negative peptidoglycan is even thinner than is drawn in this diagram. But to make up for that, gram-negative bacteria actually have an outer membrane that's external to the thin layer of peptidoglycan, and the outer membrane is still considered part of the gram-negative cell wall structure. So the two upper layers are the cell wall, and then the lower layer is the inner cell membrane. So you have the outer membrane, you have the inner cell membrane, and this entire package is called the bacterial envelope, taking into account both the cell membrane as well as the cell wall. You get the cell envelope. Well, I'll diagram cell wall and cell envelope further during our lecture portion. For now, let's just take note of the presence of the outer membrane in gram-negatives in addition to the thin peptidoglycan. So they do have an outer membrane. And if you look carefully at a gram-positive cell wall, there is no outer membrane. There's just a really thick peptidoglycan layer, and all of those layers of peptidoglycan, one on top of another, they're stabilized by the presence of these wavy lines that represent something called teichoic acids. And so teichoic acids are very important in stabilizing the peptidoglycan and also fixing the peptidoglycan down onto the cell membrane so that the two are stably joined together. And so we'll write that in addition to thick peptidoglycan, there's the presence of teichoic acids. We're going to go into even more detail in lecture about specific types of teichoic acids, such as wall and lipoteichoic acids, and we're also going to go into more detail about the outer membrane because there are many chemicals that are present in both cell types that influence how doctors are going to proceed to treat someone who has infections with either of these types of bacteria, and also because we want to know more. But for laboratory, I think this is sufficient to memorize these details. Another big detail is, at the end of the day, when we get done with our gram stain, if the cells appear pink, that means gram negative. On the other hand, if you see purple cells like we see in these grape-like clusters of staph, that's going to tell us that those cells are gram positive. So we need to memorize that. Gram negative cells are pink and gram positive cells are purple. Okay. Now, I'm going to do a close-up on the camera in just a little bit, and I'm going to show you guys exactly how a gram stain is performed. But before I do that, I want to make sure and remember to tell you right now that the gram stain, these designations, gram negative and gram positive, are not referring to the charges of cells. So I know in the previous laboratory lessons, we learned about the simple stain and the negatively charged cells and the positively charged dye and how the opposite charges attract and the dye is absorbed. And then the opposite was true with negative stains. That is not what we're talking about here. Mr. Graham, Hans Christian Graham, he may as well have said that the gram positive and gram negative cell designations, they could have very well been just called gram X and gram Y. Gram X, we could say, has thin peptidoglycan, and gram Y could have thick peptidoglycan. But that's not how the designations got started. And so we have to go with the conventions of, the way that it's referred to out in the everyday world, which is that when we say gram negative, we're not referring to charge. Gram negative is telling the physician and the pharmacy and anybody who's involved in that patient's treatment that the person that they're treating has this type of bacterial infection, that the cell wall looks like this and will respond to certain antibiotics. And by the way, it is not good news, generally speaking, when a person has a gram-negative infection. And the reason is because this outer membrane is rich in lipids. And when you have a lipid, fatty outer membrane protecting the gram-negative cell, it means that certain water-soluble antibiotics will not be able to get past the lipid layer because water-based substances and lipids do not mix. They tend to kind of go away from each other. And so many antibiotics will bounce right off of the surface of a gram-negative and not penetrate. And so gram-negative bacteria require certain antibiotics that may be different than the treatment that works for gram-positive bacteria. So it's important to remember there are many factors why gram-negative and gram-positive affects the treatment regimen, but also that we're not talking about charge. So when we say gram-positive, you're talking about trying to treat an organism with a cell wall that looks like this, and that's responsive to certain treatments, and it's not about charge. So I'm going to jot that down. You need to remember this fact. Gram, and by the way, the result of a gram stain is referred to as a gram reaction. Gram reaction, or I'll just put stain, results are describing, or I should put describe, cell wall. I'm even going to use a different word than describe. How about we put reveal? Reveal cell wall structure. Not charge. Okay. Okay. Good enough, guys? Okay, this is going to be before the 4 steps of the gram stain. We have to effectively take samples of bacteria or a clinical sample of some sort and put it onto a slide and put it through a process called heat fixing. Okay. Before we do that, though, this is part of the slide setup, but we can just take a quick glance at what are the 4 steps of the gram stain. So I'll review this later, but I just want to preface it right now. We're going to be putting crystal violet on the cells, just like Graham did. We put the purple dye on first. Then we'll be following that up with an application of iodine. Iodine, by the way, kind of just touching on charge for a moment about chemical interactions. Iodine has a negative charge. So if you've been a chemistry student, when you write chemical equations that involve iodine, you almost always write I- for the iodide ion, which is what's reactive in chemical solutions. It helps to kill bacteria if you've ever used it as an antiseptic. Iodine works to kill the bacteria, but it also has the ability to combine with the positively charged crystal violet. We know that simple stains have a positive charge, so these two actually complex together, and so the iodine acts like a fixative, or what's technically referred to as a mordant. A mordant is what functions then to keep the crystal violet locked into the cells a little more than if it was not used. And then we're going to follow up with a decolorization. The decolorization step is slightly modified from when Hans Christian Graham did his work. Graham reportedly used a 95% solution of pure ethanol and like 5% water. This is actually an acetone alcohol-based mixture, and I found that acetone alcohol, like many labs use, is much more forgiving. The amount of time that's required to apply the alcohol and acetone until the solution runs clear on the rinse has a little bit broader window to still be accurate. So I'm going to estimate that we're going to apply this for about 10 to 15 seconds. It's not an exact science. It's more of a little bit of an art here, but we don't want to go beyond this by too much. So 20, 30 seconds, a minute, that's way too long because alcohol and acetone as well. Acetone, by the way, is like fingernail polish remover, paint thinner. This stuff will eventually just dissolve away all the cellular components. And then, of course, the crystal violet will come out because you're breaking down the cell. And that's not what this test is about. This test is about a brief exposure and then just trying to see if the cells will lose the crystal violet fairly easily. If they do, then what's going to happen, we'll see on the fourth step, is that the secondary stain, which is our safranin, will do the job of colorizing those cells that lost the crystal violet. Either way, though, we always apply the fourth and final step, which is this safranin step. It's called a secondary stain because it is the second type of dye after the primary dye. So you've got your crystal violet, which is a stain, and then the safranin is a stain. The iodine and the alcohol decolorizer, those are not considered stains. So don't get mixed up, guys. I think I've asked before on a quiz or an exam or something, hey, students, can you tell me the name of the secondary stain in the gram staining procedure? And sometimes students get mixed up and they think I'm talking about the second step, but the secondary stain is actually the safranin, which is part of the fourth step. Anyhow, not trying to make a trick question, but it is called a secondary or counter stain, as it is the second dye. All right, so it's time to go ahead and heat fix a bacterial smear. We'll take a zoomed in close-up look at that. So this is a close-up of how we'll be applying the bacteria onto the slide in preparation for the gram stain. We're going to call this making a heat-fixed smear. And this is before the gram stain procedure itself. So we have to learn about this step called making a heat-fixed smear. Now, I've already pre-labeled my slide to say gram stain, and then it says CF plus SE. And the CF and the SE are the initials of Citrobacter frundii. And you can see Citrobacter growing on the slant here, colonies of creamy growth. And we'll just take a tiny little sample of Citrobacter, CF. And we'll take another little sample of bacteria. We have here Staphylococcus epidermidis, which the initials are SE, and we'll take a little bit of that and mix it in to the same puddle of water that we're going to be using, so that we can make a mixture of both. All right, so we're going to start off with the use of a loop, and the inoculating loop, as it's referred to, is a quick way to sterilize an instrument that, I mean, it's quick to sterilize this instrument and get a drop of water as well as bacteria, depending on which instrument you want to use to get the bacteria. We'll use this to get the appropriate amount of water. So I've flame sterilized my loop, and then what I'm going to be doing is using this water bottle here to get the right amount of water. You don't want to just simply spray water onto your slide. You want to take the water bottle and fill the loop so that there's a big hanging drop of water hanging off your loop. Oh, it keeps falling. And that's okay. We'll see here. Okay, so we've got a drop of water. And that amount of water that you see, it just fell. That amount of water that you saw just now hanging off was too much. And so we want to shake off quite a bit so that there's just barely the amount of water that you see that fills up the loop, if you can see that. And then I'll take this off the Kim wipe. Maybe against the dark surface you can see how much water is sitting on my slide now. The amount of water on the slide, after you set the loop down several times, should be about the size of a dime, no bigger than about the size of a nickel. If you've got a quarter-sized or larger amount of water, then you've got way too much water on there, and it's going to make this process a lot more difficult. So I recommend a small amount of water. And now what I'm doing is picking up a different instrument, looks just like the loop, but it's straight at the end. It's called an inoculating needle. And this needle will be used to pick up a very small amount of both organisms. Doesn't matter which one you start with. I'm going to go ahead and start with the Citrobacter fundii. I'll put my palm face up to the ceiling. I've let my needle cool down. I grab the cap between my pinky and ring finger, unroll the tube. Always flame your tube before you enter into it. Very carefully, I'm going to go down in there and just wipe the needle through a very small section of the growth. I should be able to see just the smallest little bit of bacteria on the end of the loop. No more than a centimeter, oh not loop, sorry, the needle. No more than a centimeter of that needle shows any evidence of bacteria, but when you rub it around or stir it around inside that little puddle of water, you can start to see the puddle of water get milky. That's called an emulsion, and it's a mixture of thousands, if not millions, of bacteria that are now present in there. Now we don't want that to dry out because we're going to stir in the other bacterium. In this case, it's Staphylococcus epidermidis. So I flamed off my loop and I make sure that it's, my needle, sorry, make sure that it's cool. And now I'm going to take my needle and go in and get a sample of Staphylococcus epidermidis. The rookie mistake that a lot of students make when they're beginning is they just pick up so many bacteria that it overwhelms the sample. We want the smallest amount. I see just maybe, I think I said a centimeter earlier. I mean like a millimeter, maybe 2 or 3 millimeters maximum of that end of that needle is covered in bacteria. Very small amount. That's why we don't use the loop. The loop has a lot more surface area, and it makes it difficult because students pick up too much with their loop. So we use the needle in most cases, and what you do is you stir, thinning out that smear or this puddle so that you can make a little bit bigger smear that takes up about the size of a quarter. Okay, so we're spreading it out. And we want that to dry all the way air dry. And the way that we're going to do that is by using something called a slide warmer. The slide warmer actually speeds this process up significantly. So I'll bring that over. Here, I'll just set it across like this. You can see the edge of the slide warmer in your picture now. And what I'm going to do is just, oops, there we go. The slide warmer says that it's at 45 degrees Fahrenheit. That's pretty hot. You know, it's not going to burn you, but that's toasty warm. And so 37 is human body temperature, and it's at about 45. And pretty soon, you can actually see that the smear is turning like a dry, chalky white. And what's happening is all the water is evaporating and the bacteria are drying up. But that doesn't mean they're dead. Please realize that you need to wait until all the bacteria have completely dried up. And even then, if you add water back in, they're still alive. Unlike animals, such as a human or some other higher form of life like higher form than bacteria, when you dehydrate most organisms, they die. Bacteria are pretty hardy in that most of them, if you just add some water back in, they would actually still be alive if you just simply rehydrate them. So I'm waiting patiently to make sure that every part of that is bone dry. And the reason is because now what I'm going to do is grab a slide clip and do what's called the heat fixing step. So this is all the way dry now, and we're going to put the slide on the slide clip, and we'll go ahead and light the Bunsen burner back up. We'll turn it down a little bit. We don't want it to be too flaming high. And what we're going to be doing is actually passing the dried bone dry heat fix, or bacterial emulsion smear through the flame 3 separate passes. And you want to do this carefully. If you go too slowly and you start to see this smoking, that's a really bad thing, because smoking bacteria is bad. Smoking is always bad. But yet at the same time, we want to do this slowly enough so that, that the bacteria do actually get killed by the flame. Make sure you have the slide on the right side up, meaning that the frosted labeling area is face up and that the bacteria are on top of the glass. If you accidentally flip it over, then you're going to incinerate the bacteria, and that's no good. That'll ruin your sample. So here we go, guys. Watch this. I'll approach the flame right at the tip of the inner cone, slow down over the flame, and come off the flame. You never stop over the flame. Okay, you approach the flame, slow down, come off the flame. And for one last third pass, slow, come off the flame. That is a good heat fixing technique, and we can set that aside, turn the flame off. You don't want to do any more than that. We're not going to add a cover slip. The gram stain is actually performed right after this. So let's go ahead and see the gram stain performed next thing. Let's review the way in which we produced the heat fix smear. So we now have our heat fix smear, but what I want to do is just clarify a couple of the technical details that are behind the, why we perform these steps. So let's see. Why do we heat fix? Why make a heat fix smear? And so let's look at the purpose first of all. Running the bacteria over the flame carefully, gently, accomplishes more than one outcome. So it's multi-purpose. First of all, it kills the bacterial cells. And that's good because we don't want potential pathogens hanging around where if you're processing a sample that you would come into contact with them potentially. When handling clinical samples, you would be wearing gloves and you'd have personal protective equipment, PPE of all different sorts depending on what's going on. Masks, there can be biological safety hoods where the air is sterile filtered. And in this case, I was very lax because I'm using harmless, non-pathogenic bacteria. So, but ultimately, I'd still like the bacteria to be dead before I do any other steps. And so first we have killed the cells, but not incinerated them. They're not burnt into a black crisp or smoking or anything. They're just gently killed. Cue up the music. Killing me softly. All right. What else? Kill cells. Oh. The cells will stick down onto the slide. So it sticks, or another word would be adheres. Cells to the slide. And thirdly, we have the fact that when you perform the steps of a heat fix smear, you now have a slide like this one here that'll last maybe a month. That's a long time that we could wait. Like let's say we ran out of time right now, we could just skip doing the gram staining application of the actual chemicals, and we could wait until next week or maybe next month, and that's without a cover slip or anything. So preservation is an important point. It preserves the cells. how are all these things accomplished? Why do the cells die? Why do they stick to the slide? How is it that they're preserved and that they're not going to rot or decay in some way that, you know, might otherwise happen? And all of this is accomplished through a mechanism of what we call protein denaturation, the way in which the proteins that are inside the bacterial cells and on the bacterial cell surfaces, they denature in such a way that they coagulate. Now, denaturation means that these proteins, when they're heated up, they change their conformation so that they're no longer functional in a native sense. When proteins are in their native conformation, they will continue to function and the bacteria are alive. But when you heat them up, they coagulate, and we call this protein denaturation. Or coagulation. Specifically, that's how they're denaturing. And maybe the easiest way for you guys to think about a proper heat fixing technique is that it's like cooking an egg. Imagine heating up a pan, nice and hot, cracking the egg in there. When you crack a raw egg into a frying pan, it starts out that the egg white part is actually clear, right? All the proteins, the albumin that makes up the egg protein, that's all clear and colorless. But when it hits the hot pan and you cook it just right, the egg white will turn white, the yolk will be in the middle, and that's fine. You've got a nice cooked egg. But if you left it on there for too long, then eventually the fire of the stove would just heat the pan up to where it would turn black and it would start smoking and incinerating. And it's the same thing here, guys. We're trying to make sure that the egg is fully cooked, or the bacteria are essentially dead, just like the chicken egg or whatever egg you're cooking isn't going to grow a bird anymore. It's denatured, but it's not cooked to a crisp to where you've got some incinerated black thing. So realize that protein denaturation is very much the same thing. You're cooking the egg or the bacteria, but only to the point where the proteins coagulate and are not completely destroyed. The next thing that we're going to do is look at performing the gram staining technique. I'm going to go ahead again and put this on a camera close-up so that you guys can see each chemical as it's applied to the slide. And so let's take a look at that. So here's our close-up view of the gram staining technique on heat fix smear that we just prepared. So we have our heat fix smear. You can see the milky white dried out bacteria, and they're dead already. But there has been some debate as to how effective is the heat fixing technique. So we did the gentle heating and it was only 3 passes. How can we be sure that all of those bacteria are all the way dead? And the answer is that they might not all be dead, but most of them are. And so just to be extra certain that they're all dead, we're going to use our first step of the gram stain, which, if you recall, is crystal violet. Once the crystal violet is absorbed by the emulsion, this heat fix smear, the bacteria will certainly be dead. And interestingly, I had a student one time who said he was from Ghana, a country in Africa that uses crystal violet as a type of antiseptic. Apparently, due to its antibacterial properties, it is, I guess, a means of preventing infection in cuts and scrapes and such. One of the reasons why you don't hear much about crystal violet being applied here in the United States as an antiseptic is I think it's just really messy. But I'm applying this all over my heat fixed smear now for that step 1. And we're going to do this for 1 minute. So I just looked at the clock and I'm waiting 1 minute to make sure that every bit of the bacteria on the slide will have picked up the purple dye. And I need to be prepared. Look at the color of that clothespin. I've been using this as a slide clip because I don't want my fingers to look like that. And so at this point, whether you're talking about the Citrobacter frundi or the Staphylococcus epidermidis, all of the bacteria are going to be purple. So whether they're gram-positive or gram-negative, they'll be purple. All right, so I'm going to get ready to dump off the crystal violet now that the 1 minute has passed. It's almost been 1 minute. I'm trying not to rush it. You want to make sure you go the whole minute, but you also don't want to walk away and answer the phone or do something else that's distracting, because once that 1 minute passes, you want to get that off of there. Crystal violet will eventually start to crystallize, and it's difficult to rinse off. So we don't want that to happen. This is my rinsing technique. I'm using a rinsing bottle, just a little spray bottle here to get a fine stream, and you can see that my technique uses the slide at an angle. Another alternative would be to actually turn on a soft stream and just rinse that off gently. What we don't want to do is use the rinsing bottle like you know, blasting the bacteria. If we're blasting them off of the slide, then we could do damage to the smear. Realize there is no cover slip, and you want to do that gently. So sometimes students ask, do I need to dry the slide in between applications of chemicals? And the answer is no. We can just shake off the excess water. And if you recall, the second step of the gram stain is grams iodine. When we apply the grams iodine, we're going to put a generous amount on there. You always apply these chemicals until every part of your bacterial smear is covered. So we've got a nice puddle of iodine, and if they weren't dead before, now they're really dead, because as they absorb the iodine, that also has an antiseptic property. These chemicals just tie up the cytoplasmic contents and the proteins and everything. If they weren't denatured before, they certainly are now. And so iodine, what it's doing, if you recall, it's combining with crystal violet, complexing with it, so that the crystal violet is that much more locked in or fixed into the cells. So at this point, all the bacterial cells are still purple, but they now have iodine soaking in them, complexing with the crystal violet. Iodine has a minus charge, the crystal violet has a positive charge, and so the two complex together to make what's called the crystal violet iodine complex. Now it's been 1 minute, and I'm going to go ahead and dump off the excess iodine and just give it a gentle rinse. Make sure it's all the way rinsed off. And now it's time to do the most critical part of the gram staining procedure. The most critical part is the application of the decolorizer. In this case, we're using an acetone alcohol solution. And the reason is because we're going to apply this dropwise until the slide has clear runoff, meaning that no more purple dye will be coming out of the smear. But we don't want to do this for more than about 10 to 15 seconds at the maximum. It depends on how thick your smear is. I'm guessing my smear is kind of medium to, it looked a little thick. So we'll see how this goes. Be prepared to rinse this off though as soon as your 10 or 15 seconds is up. And that's because you don't want it to stay on there for too long. So I get a dropper full, hold the slide at an angle, and you should be able to observe on camera right now the purple collecting at the bottom. You see all that purple collecting at the bottom? I'm going to get another dropper full and continue this process. Okay, I'm not really seeing much purple at the bottom anymore. It's been the 15 seconds or thereabouts. So I'm going to go ahead and gently spray that off, rinse that off to stop the decolorization from happening. It's complete. And now we have to do the fourth and final step, which is to add the secondary stain, also referred to as safranin. And it has a light reddish pink color. And the longer you leave the safranin on for, the bolder the color starts to become. We really would like the safranin to colorize the cells to the level of kind of a light frosted pink. And so that's going to be 1 minute. You definitely don't want to go more than 1 minute unless your safranin isn't staining as expected. The level of safranin staining should be about correct at 1 minute or less. But I wouldn't go, I just would stick with 1 minute. And at this point, realize all that purple dye that you saw running out to the bottom of the slide, that's gone out of the cells that decolorized, namely the gram-negative cells. Gram-negative cells do not hold on to the purple crystal violet staining, and so this safranin is being soaked up by all the cells, but the ones that you'll notice soaking it up are going to be the gram-negative ones. They were clear, and now they're like a light reddish pink. Okay, so we dump this off after 1 minute, and we follow it up with yet one last final rinse. And you want to make sure you get all the dye off of there. It's almost just easier to use the sink for a lot of these applications, just to make sure that you don't get the dye on your hands. And then you can just take a Kim wipe and wipe the bottom and gently blot the top. And then this can go under the microscope, and I'll give you guys a close-up view through the microscope of the cells on here that there should be some purple and some pink, because we've put CF and SE are actually both gram-positive for the, what is it, the staph cells, SE, and then the CF is gram-negative, so you'll see pink and purple in this slide. All right, so that'll be the next thing that we'll take a look at. And here we are looking at a close-up microscopic view of the gram stain. So this is at low power, which is 100x, and it's not zoomed in enough, not magnified enough for you guys to see the shapes of the cells. What we're supposed to see at this point is that we have our smear in focus, and you would want to make sure that you can see a little bit of empty space in between some of the cells on the smear. So we can see a little bit of glass in the background and all those little scatterings of what look like purple and pink dust are the bacteria themselves. And I'm going to leave it here and put a little bit of oil onto the slide itself and switch over to our maximum level of power. And because our scopes have the parfocal engineering, just a little fine focusing, and we can see a close-up now. We're looking at 1000x maximum magnification. And if we zoom in as fully as we can here, it's a little shaky, but we should be able to see the pink cells surrounded by some purple cells that are in the grape-like clusters. And it's a little hard to see, but the pink cells, which are the lighter colored cells, actually have a little bit of an oval to short, fat rod shape about them, like the one at the end of the pointer there. That's actually referred to as a coccobacillus. Now, if you look at the purple cells, you can tell that the purple cells are definitely spherical, like typical staph cells. So the Staphylococcus epidermidis are the purple clusters, and then the pink cells really don't have a particular arrangement, but they are the gram-negative Citrobacter frundii. Remember, purple is positive for the staph in this case, and pink is what we see for the Citrobacter gram-negative cells. And that's as much as we can get out of our gram stain right now. Turned out to be a pretty good result that I just made. Oops, keeps going out of focus. Right, you just find focus. And there you have it, guys. Now that you've seen the gram stain under the microscope and you've seen how it's performed and how to make a heat fix smear, etc., keep in mind one thing. This also employed some of the uses, some of the steps that are used in performing a simple staining procedure. So the first time I did a simple stain, I swabbed my cheek cells and I floated them in what's called a wet mount technique, where I put a cover slip over that, and it was very temporary. They dry out and then they're no good to look at anymore. They're not put under like a long-term preservation cover slipped process that can be viewed other than just right then and there. When you want to do a preserved simple staining procedure, you could just basically do the first step of the gram stain. So you could make a heat fix smear of whatever it is that you want to observe, and then just simply add whichever primary dye you'd like. So you could, whatever you're in the mood for, some purple cells, add the crystal violet, or if you wanted to use methylene blue like I did on my cheek cells, or saffronin to make them reddish pink, or you could even do malachite green, which we haven't used yet. That would be performing a simple stain. Just add the dye, soak it for 1 minute, rinse it off, blot it off, and you can look at it under the microscope. Keep in mind though, if you only do step 1, which is the simple stain, you're not really getting the gram stain information. The gram stain goes through all 4 steps required every time, and they have to be done for the right amount of time. And then the information actually tells you about cell wall structure on the purple versus pink outcome. And you must use the particular chemicals. You can't just put, for the crystal violet, you can't just swap in malachite green. It doesn't work that way. All right, so you have to do everything precisely as we've done there. And let's review now. One of the review sites that you may find on the internet, I put here currently unavailable, but we'll see if we can get this to work. I actually have downloaded this from this site. If you try it, I've downloaded it, and it's basically a tutorial on how the gram stain works in like an animated video that you can click on. It's interactive. And so we'll use that for our Zoom sessions. I think we can fill this in together though, which is just a little way of quizzing you briefly. Do you understand what color each cell type is after each step? So if we add crystal violet, then of course the purple dye will get absorbed by both cell types. So we have a purple-purple situation here. The second step of the gram stain, using the iodine, that just enhances the purple dye coloring and fixes it in. So once again, both gram-positive and gram-negative cell types will remain purple after this second step of the gram stain. The most critical step of the gram stain, which is the acetone alcohol decolorizer step, if it's applied properly, we will see that the gram-negative cells with their thinner peptidoglycan cell wall will actually lose the purple coloring. If they lose the purple coloring, they'll go back to their original uncolored state, which is to say clear. Gram-positive bacteria with their thick peptidoglycan will actually retain the purple color. And we've got to do this correctly, otherwise there may be issues with what we call over or under decolorizing. And I'll talk a little bit more in detail about how if you over decolorize or you under decolorize, that can have drastic consequences to the accuracy of the gram stain. And finally, the saffronin, when you add a light reddish pink dye on top of these cell types, the purple cells are already deep purple. Think about what happens when you add deep purple, you add light pink saffronin on top of an already deep purple cell. Nothing. It'll still look dark purple. And on the other hand, the clear cells will have soaked up the saffronin, and they will become pink or red. So you'll have purple. Gram-positive stay purple the whole time, and then the gram-negative cells go through this transition and end up pink. And there you have it. Before we move on from this topic, let's talk about what happens when this most critical step is performed badly. Maybe a student falls into memorizing this wrong and they say, oh, well, this is 1 minute and this is 1 minute and this is 1 minute. If you did the decolorization for 1 minute, then you're going to pretty much break up the cells to the point where they'll lose the crystal violet no matter what, and everything will have turned clear, even the gram positives. And so what do you think the consequence would be there? I'm going to go ahead and put this on its own slide. I forgot to add this in. We need a slide that is entitled Accuracy, Factors that Affect the Accuracy of the gram stain. Okay? Factors affecting gram stain accuracy. Okay? So I think what we should do is let's digest this topic during our next Zoom meeting or when this comes up in our Zoom meetings, and we'll go back through the steps and then we'll think this through together. But be prepared. A big part of this is going to be the over or under decolorization as a driving factor if the alcohol is applied for too little or too long. But there are other factors as well, which is how long can these bacteria remain healthy in their test tubes? How were they incubated? And are they overly crowded? Have they run out of food? Do they have perhaps a problem with dying of old age? And all of these things could be a driving factor then in what's going on with accuracy. So we'll detail that out during our Zoom discussion. And that brings us to our final topics for the day. I'm going to flip to the, I've got to flip past a few slides here. We're going to talk about the oxidation fermentation test, just how it's set up, and also the phenol red broth, how is it set up. And my recommendation would be that you read about these, just the brief little introduction in theory sections in your lab manual. And then after the setups are incubated, we'll come back into lab and we'll be analyzing these for most of our lab period during our next lab video. So let's start with the setup of the oxidation fermentation test, OF glucose test as it's sometimes called. So for the setup of the oxidation fermentation test on the first day, we'll just take a quick look at the purpose. We're going to be testing various bacteria, including these 3 species that are listed here. E. coli, which we know is normal flora in the intestines, but there are some bad strains of E. coli that we want to be able to identify whether we're looking at a clinical sample or in the lab. This is one way that we can start to differentiate various E. coli from, say, other organisms such as Pseudomonas aeruginosa. Pseudomonas is a real problem for people who are immunocompromised, like maybe someone with severe burns or like a burn victim, someone who, say, gets in a motorcycle accident and a large part of their body is just, taken right off in terms of skin. And Pseudomonas is one of the biggest threats. And so although it's safe to handle in laboratory and everyday settings, when it does get into people who are in these situations, very specific medical situations, it can be a real problem, even deadly. And Alkyligenous faecalis is yet another organism. These are all gram-negative organisms that we'll be comparing. Alkyligenous faecalis is a normal flora of the gastrointestinal tract, like E. coli, and it generally is non-pathogenic, but it sometimes pops up when bacteria from the GI tract get in places where they don't belong. So we're going to be comparing these 3 species through putting them into 6 different test tubes. Now, why 3 organisms, but in 6 tubes? And the setup basically requires that each organism be put into the same type of media, right? This is what the OF medium looks like. It's a green jelly-like agar deep, but one of the 2 test tubes, after it gets bacteria, will also get a layer of mineral oil. So what you're seeing up top here is a fairly thick layer of oil that blocks out access to oxygen. And so we're going to do this for each one of the organisms. We'll have an anaerobic with oil and an aerobic without, anaerobic with oil, aerobic without, for each organism. And I'm going to go ahead and just set up one organism, for example. So I'll take some E. coli and put it on the end of a needle and stab it down into the test tube. And before I do that, I want to make sure you guys understand how to effectively stab the bacteria down in there. We only want to do one nice steady stab, not multiple stabs. And if this is your test tube, the stab, one second here, I'm going to redraw that. The stab down into that deep should only travel down about just short of 1 centimeter of the bottom. So we don't want the needle to touch the glass at the bottom of the test tube. Whenever stabbing an agar deep, putting the bacteria in there, you always stop short of the bottom. And I'll remind you that in other experiments as well. So we get out this instrument called the inoculating needle, and we turn on our flame. I always listen for the gas pressure. You just want to barely hear that hiss of gas. You don't want it on too strong. And we have to have our bacteria ready to go. So I've got some bacteria, some E. coli that I can test. And what I'm going to do is just pre-loosen the cap so that I want to make sure that, that it's easy to get off there when I'm ready. And then you can label some test tubes EC for E. coli, as well as your initials on there, so we know that you made those versus other students' test tubes. And here we go. So we're making this setup right here. I'll flame the loop. Oh, not a loop. I keep saying loop. Needle. Okay. Once the needle is sterilized, we wait for it to cool down, and that gives us a moment to get our bacteria. You put your palm face up towards the ceiling, and you grab the cap carefully between the pinky and ring fingers so that your skin does not touch the bottom edge of that cap. And you don't want the cap down on the counter. Recall that this needs to stay out of the way and face down so that contamination from the air can't fall into the cap, and it's out of the way. We're going to flame the tube like we always do before we go into a tube. And very carefully, I will reach in, set the needle down on a cool part of the agar, make sure not to burn the bacteria, and then I'm going to wipe my needle through the bacteria. I don't even see any bacteria on the end of it. Do realize that there are still invisible amounts that are hundreds of thousands, if not still millions. Now I'm going to grab this cap on this brand new OF glucose agar deep, flame it, and then I'm going to hold the needle steady, and I use my right hand to direct the needle down into there without touching the sides. And I go down almost to the bottom and come out nice and steady, sort of like a drill press. You just want one nice steady hole right through the middle there. And now that's finished, and we're going to flame off the needle. And we'll just repeat that process one more time. So we get the same bacteria, put your palm face up, grab the cap, unscrew the tube, flame it. And this goes more quickly as you get used to doing these steps carefully and without mistakes. If you make a mistake, it's not a big worry. You just have to be aware of it. Make sure that you start that tube or that step over with. Make sure that you fix any mistakes that you made. Another way to grab caps is rather than between the pinky and ring finger, you can do the pinky wrap on larger test tube caps like this one. I did the pinky wrap just now. And now I'm going to go in and carefully make that same stab, but in a new test tube. So I've got my second OF tube. There's a little bit of bacteria left on the end of the needle, but that's okay. Whatever came off of the needle during that one single stab, that's plenty. And then make sure that you flame off any excess bacteria. So, bye-bye. Sorry, guys. Sorry, not sorry. I don't really want those around. You know, I have to say, if torturing bacteria or mass killings is a crime, I'm going straight to hell. I'm glad I don't work with, like, you know, laboratory animals and such. By the way, what I'm doing right now is I'm putting on this fine layer of mineral oil, or I shouldn't say fine, a thick layer of mineral oil. So I've got some sterilized mineral oil that I'm going to go ahead and just very carefully hover over the top of the test tube that I've already inoculated. I've already put bacteria in there, and I put a nice big thick layer of mineral oil over that. Notice that I hovered over the top of the test tube and I did not place the sterile mineral oil dropper down into the test tube. Think about why. You want to make sure that no contamination from the bacteria in that test tube gets into your sterile mineral oil dropper. Otherwise, and I've seen it happen, students will then pass this to someone else, and a whole bunch of people will have some kind of contaminant in there that gets into everyone's experiments. And a little bit goes a long ways. You know, 1 million bacteria can fit on the head of a pin, and so contamination can go through a situation very quickly. Okay, so at this point, these test tubes are going to go into the incubator, and we'll take a look and see what they look like next time we come to lab. Our final setup for the day is going to be the phenol red test. The phenol red broth, or PR test, PR broth, is one that evaluates a bacterium's preference for sugar or not. Will they digest a specific sugar? So we can take sugars, common ones, like let's take glucose for instance. Here is a phenol red broth with glucose sugar in it, and we're going to use a loop for this particular procedure. You could use a needle. It'll probably work just fine too. I've used needles before for this, but traditionally we use a loop and scoop up one of 4 different types of bacteria that we'll be testing. So generally students will work in groups of 4. And the E. coli would be assigned to one student, and then another student will take Enterobacter aerogenes, which is another fellow gram-negative bacterium that lives in the intestine as normal flora. You've got Alcaligenes faecalis that I mentioned in the previous test just now with OF. We'll be testing that one. And then here's a new one, Proteus vulgaris. This can be abbreviated with the initials PV, and this is also found in the intestines, but its name is special in that it was named after a sea god, Proteus. Apparently, this was in reference to the ability of the organism to swim really well, like massive numbers of flagella on its outside bacterial body, and it allows it to propel itself through the intestines really well. These organisms can also be motile, but motile meaning that they can use flagella, but Proteus is known as being exceptionally good at that. So what I'm going to do at this point is just tell you that the setup here that we'll be analyzing is to take all 4 organisms and test them. So E. coli will be tested for a phenol red with glucose in it. And then we'll also test E. coli in a phenol red with lactose in it. And the way that we differentiate between them is all the broths are red, but the ones with lactose will have a metal cap, and the ones with glucose will have a plastic cap. And also, the glucose phenol red broths have a little extra upside down test tube added, a mini test tube that's added into the broth, and that's called a Durham tube. And we'll talk about how that's for gas collection readout when we get back next time. So what you guys are going to be doing is using the inoculating loop in this case to make this happen. So to make these setups possible. This is really easy. We don't really need a close-up view. I'm just going to turn on the gas one last time before we call it a day today, and that's so that we can get a little sample in the end of our loop, whatever organism you're testing. Okay, so I'll take this tube of bacteria, I'll put my palm face up, grab the cap between my pinky and ring finger, carefully unscrew the cap, flame it. Business as usual here, right? Don't touch the sides. Go in there, cool that loop or that needle off. In this case, I've got my loop, and I'm just wiping it through a little bit of the bacteria. I can see the tiniest little bit of bacteria on the end of the loop right now. And what I'm going to do is put it into this fresh phenol red broth. So I'll do the pinky wrap this time. Grab the cap. Now it's out of the way. Flame that test tube. Stir these bacteria in. Just shake it around. There's no real special technique here. We're just trying to make sure that the bacteria will get off of that inoculating loop. Even if there's a little bit of bacteria left on the end of the loop, it is not a problem. Plenty. You have to trust that plenty came off and into the phenol red broth. So we flame that one more time. We put the cap on. Always make sure that you fully flame off your excess bacteria and anything else there on that loop. And now that's safe to set down. Always turn your flame off after you're done so that you don't have a dangerous open flame just blasting away. Okay, so what we're left with is that our samples that we've set up, and for an entire class set, you're going to have all 8 phenol red broths and all 6 of the OF tubes. So we'll be analyzing 14 different test tubes from 2 different testing methods, OF and phenol red, and we'll see what the results mean when we come back to lab next time. And that's it for today. Thanks for sticking with me on a really long analysis. Gram stain was a really big one for you guys, and I hope you guys all learned a lot from today's activities, and we'll be reviewing them in our Zoom meetings. So I'll see you next time. Thanks.